Welcome to today's intimate conversation with Cami Think Tank. My name is Kamini. I'm one of the co-founders of Cami Think Tank. The other co-founder is Michelle Yee, who is also under the Kamini Gupta tile, so just ignore that. But Michelle, give a wave to everyone. Say a quick hi. And we started Cami Think Tank about a year and a half ago now, when a lot of our former clients came to us and, with the Gen AI hype cycle and whatnot, they're like, how do I actually use this in practice? And can we leverage this within the life sciences area? And so that's how Cami Think Tank came to be. We are an education organization that provides advisory, workshops, and just overall education for scientists and folks who work in pharma and biotech to just up level on how to best utilize AI within your work streams. And we have a self-learning membership platform, which I know a lot of you have joined already. So thank you to all of you who have joined. And within there, you can get classes, attend some, workshops that we might be hosting later down this year in person in San Francisco. And then also we have a number of role-specific guides for folks that are just looking to upskill, up-level themselves within the AI area. And lastly, we are going to launch a podcast in March. So that way you don't have to read through all of the guides that we have. You can just listen to the fun information there. And so just keep an eye out. You're likely already on our newsletter, but keep an eye out. We'll be releasing those in March, and excited to have you here for the journey. Now, without further ado, I would love to hand it over to our guest, Rachel Thomas here, who we have the distinct honor, honestly, of hosting on this call. So Rachel, thank you so much for joining. And Rachel has been within the AI world for quite a while. She'll tell us more about that. But she also knows a lot about science and a lot of the risks and limitations of AI within life sciences. And so with that, I'm going to hand it over to Rachel for a quick introduction, and we'll jump right into the questions from there. Go ahead, Rachel.
Thank you. Yeah, and thanks for having me. I'm really excited about this discussion. So I'm Rachel Thomas. My background, I earned my PhD in mathematics, and I actually did some biology math modeling as part of that with medical applications. Then I worked in the tech industry as a software developer and data scientist. And then in 2016, I co-founded Fast AI with the goal of making deep learning more accessible and easier to use for people kind of from different domain areas, including biology and the sciences. While working at Fast AI, I was also working at the University of San Francisco, and I became very, very interested in ethics issues. I helped start a Center for Applied Data Ethics there. And then I went on a few years ago, just became increasingly fascinated with the life sciences, and I went back to school for a master's in microbiology and immunology. And this was really fascinating, and I did focus on how AI is being applied to these areas. And so this was kind of an exciting, with this topic and in our discussion today, to kind of pull on my previous and ongoing interest in AI and its ethical use, and particularly in applications to medicine, biology, immunology, kind of all these life sciences.
I love it. I love it. And Rachel, the way that I came to know of you is actually through your blog. You wrote an amazing, amazing piece about enzymes, and how a really popular paper in Nature actually, did not have the correct, perhaps, information within it, or it wasn't replicable. And I know we're going to chat about that later, but that's how I got to know about the work that you were doing. And it was really, really interesting. So for anyone who is interested in Rachel's writings, I've just posted her blog onto the chat. So please check it out. I also love the style in which you write. It's not very boring or dense. It's fun, and it makes fun of, I think, a lot of the hype cycle that surrounds technology. And I love that. So thank you for putting your thoughts and writings onto the internet. So that way I have been able to, meet you and
Oh,
we can.
thank you. Yeah, no, this is always the goal is when I'm writing is I hope I can connect with kind of like-minded people or, get this kind of exchange of ideas. And so this is, yeah, kind of the ideal case of writing as a way to connect with people.
I love it. Well, so the first question I have for you is, we are seeing AI being applied quite a bit throughout the life sciences today. And one thing that Michelle and I talk about a lot is there's a lot of hype, and then there's the actuality of what's being built properly for AI within the life sciences. So the question I have for you is, where do you think confidence is running ahead of actual scientific understanding? Because to me, right now, it seems like building a flashy deep learning model is getting a lot more attention than the slower, quieter work of checking whether that work or the answers that that learning model is producing are actually correct. So yeah, where do you think confidence is running ahead?
Yes, yeah, no, this is a big issue, and I should say, like, I'm excited about AI. I work for an AI startup, but as you said, yeah, I do think that the confidence and the hype is often outpacing the reality, and there's also often this real imbalance. And so one big issue I see is often I feel like there's this assumption that kind of we already have all this data, and so all we need to do is like throw it in an AI model, and then we're going to get these amazing outputs. And something that really worries me is the ways that I think we may be underinvesting in thinking about new types of data that we need.
Mhm.
And in many cases, kind of the type and quality of the data you have really sets limits on the quality of the results and the work you're going to produce. And so I think with medicine, I've seen this assumption of like, oh, there's so much data in patient records. We just need to get all these like EHR, the electronic health records into AI systems, and we're going to have all these breakthroughs. Whereas I actually think that in many cases we need new types of assays. There are new biomarkers we haven't discovered. There's still a huge need for kind of benchtop research and lab research, and really thinking about new types of data, more accurate and better types. And I'm worried that that is not getting the funding or support that AI uses are. And I think that, AlphaFold is a huge success, probably the biggest success. And it's really impressive. I'm not at all taking away from the work there, but I think people sometimes lose sight of the fact that, the protein data bank is the protein data bank and the CASP competition are what made AlphaFold possible to, have this very, very well thoughtfully curated collection of data. And they started building the protein data bank in the 1970s, like on magnetic tape that they were sending through the mail. And so it was such a rich data source. And then the CASP competition was very thoughtfully and well structured. And that competition had been running for, like 20 years. And the innovations that the AlphaFold team came up with are truly impressive. But they needed that kind of the right type of data and, the high quality, and that it was such a good fit for the problem they were working on. And I think in many cases, the data is not always the right fit, and people are like, oh, well, this is the data we have, so let's go for it. And so that's one concern I have.
Yeah, and I love that you bring up that example because as we know, funding for the CASP program actually almost ended last year. And it was only due to folks really calling out how much of that program, plus the protein data bank, but that program allowed for something like AlphaFold to be created. I mean, think about how many scientific discoveries would be, at loss if we didn't continue funding for those kinds of programs. And so I think that's a really, really important call out. And it's really important in how this is decades of work in the making. It's
Yes.
not a company
Yeah.
that was just spun up like 2 years ago that did incredible work, right? So I think one other thing that you just brought up, and something that always strikes me whenever Michelle and I are taking a look at these models, is that it's really hard to evaluate these models without that deep biological expertise. Like from the outside, metrics can look really, really strong, but that doesn't actually necessarily mean that the biology makes sense. And so a question for you is, when AI systems in biology are wrong, who usually discovers that? And like, where does that ownership then lay for these new AI systems that are being built today?
Yeah, no, that's exactly the right question to be thinking about, because that's so important. And so you brought up this case study in the intro that I wrote about of this enzyme classification paper, which I think is really a important study to think about. And this is so this was a paper. It was published in Nature Communications, which is a very well-respected journal, where the team used a data set of 22 million enzymes, and they were trying to go from the sequence of amino acids to predicting the enzyme commission number, which tells you the function of the enzyme. And honestly, if I had just read this paper on its own, I would have thought, okay, this seems sound. Like they kind of did all the things that you would want and expect in this type of research, where they, had training, validation, and test sets to validate their model on this, 22 million 22 million enzymes in the data set. They also they used attention and identified regions that were known to be kind of catalytically active or important. And so then kind of once they had their model, they applied it to 450 enzymes where the function's not known and published these as novel results. And they did check 3 of those in the lab. And so what happened is a microbiologist named Dr. Valerie de Crécy-Lagarde, who was not involved at all in this research, she happened to one of the enzymes they wrote about. She had studied in the lab for over a decade. She had very deep knowledge of this particular enzyme. And so she recognized reading the paper, hey, this is false. I've already proven in the lab that this is not the function of this enzyme. And so then she dug into the other results and found that there were hundreds of errors. And so 135 of the enzymes actually weren't novel. The information already appeared in Uniprot. But the issue is, Uniprot things can be stored in very different fields, and so it can be kind of tough to sometimes. I mean, Uniprot's a great resource, but to process it. So there's this very significant data leakage. Then of the other results, there were some that were blatantly, not implausible. So they said that one gene was used for mycothial synthesis, but E. coli doesn't synthesize mycothial. So it's just okay, this is impossible. There were a lot of really highly repetitive results. They found 12 different enzymes that they said had the same narrow function. And these all point to, so I think data leakage is a huge thing that happened here, but also probably some level of overfitting to kind of get that level of repetition. But this is something that never would have been discovered if, de Crécy-Lagarde hadn't happened to read this paper and have this deep expertise. And she talked about this on a podcast. She actually had a lot of trouble getting her rebuttal to the paper published. So she tried contacting the authors, she contacted Nature Communications, she got a team together, and, wrote this other paper, and they went through multiple rejections. It was a very long process to get this published. And that really shows kind of these publishing incentives where, the paper with the exciting AI results can be published in the prestigious journal, and then trying to refute that is a much more difficult process.
Wow, that's crazy to think about how difficult the process was to refute, a lot of the claims that were made in the paper. And I think that brings up a really important point of then who is owning the fact that these mistakes were made, right? Where is this ownership going to lay? And it's something at least we're seeing time and time again as folks are starting to leverage AI within their workflows. The question that is being brought up a lot of times, from the FDA or other governing institutions of okay, let's say something goes wrong. Well, then is it that the ownership of this mistake lies on the company that may have provided the AI model? Does it lie on the company that utilized the model? Does it lie on the governing agency that might have assessed it? And I would love to hear your thoughts. Where do you think ownership should lay for something like this?
This is so tough because it occurs at so many levels. I think this case study really points to the need to having a deep integration with domain experts in other fields, and so really, closely involving microbiologists in working on this. This also points to just, the incentives of the field as a whole are not, they're not rewarding this type of work of error checking. And so it's not prioritized, and it falls through the cracks. But it is, I think it is hard to prioritize it when there's not the funding for it, or the support, or even the recognition of like, hey, this is a paper that is either the paper refuting it. I was like, hey, this is a really exciting, fascinating paper that I think should be rewarded. So that's tricky. Also, some people pointed out in discussions about this that it is, I think, in many ways, researchers, in terms of coming up with a training validation test split that truly avoids data leakage. Like, that's a really tricky thing. And so that's the thing, we saw with the CAST competition, where you have a committee of people that were really putting a lot of effort and had funding and support to do that, to create that. Whereas a lot of papers and individual projects, it really is just the kind of like individual team that may be quite under-resourced. And again, these are topics that don't get as much consideration as the actual, like, the architecture of the models, the exciting part. And so that's difficult, where there aren't the incentives in place.
Yeah, no, and I think you answered the next question that I was going to ask you, which is, what incentives in AI research or deployment worry you the most?
Oh, yes, and I can add.
Yeah, please go ahead.
I did this discussion about incentives. So a paper I love, this is from several years ago. It's called "Everyone Wants to Do the Model Work, Nobody Wants to Do the Data Work," which is just a great title. And I think most of us that have worked in data science can relate to this too, of just how much model work is rewarded compared to data work. And this paper is really fascinating. The researchers did interviews with, it was over 60 machine learning practitioners across 3 different continents. So they got really interesting variety of people. And they talked about data cascades, and it's this idea of what can go wrong in high stakes machine learning applications. But they were doing these really kind of interviews with the people at various points, and so it gives and they have like a whole taxonomy of what can break down. But you have examples of someone in the field is collecting data, and sometimes that's using sensors. Sometimes that's one example is around poaching, and so they have like anti-poaching teams, who are uploading like geographic location. You have nurses collecting information, and in so many cases, people were asked to collect extra data, but they actually weren't really given extra pay or extra time to do so. There was a lot of miscommunication where, people in the field are using like one measurement system, or sometimes they even recalibrate or change their measurement system, and then that doesn't make it back to the the computer lab where people are building the models for this data. So there were a lot of communication breakdowns. And it really, I think it's a really, really helpful paper to read about kind of all the things. And people lose months of work over this. And you have cases that they talk about where a model, so there was an anti-poaching model that when they got to deployment, basically the anti-poaching teams were just like, well, we know these results are incorrect. And it was trying to predict, where the most effective locations for them to go to. And it turns out there were issues with the underlying data set. And again, if you'd had kind of more integration of the different roles earlier, that could have been prevented. So a lot of it is really to make sure you've got those, collaboration throughout the process, people really collaborating closely across teams. And I know cross-disciplinary work can be quite difficult, and then really communicating clearly about the different steps, and then also providing the support.
Yeah, no, that totally makes sense. And what I'm hearing from you is, having those teams as they're working together, both the model team and then the scientists that are, working hand in hand, having them align on like perhaps the benchmarks that are being created, or like the safety mechanisms that need to be put in place, like really aligning them on the same vision and the same goals and the same standards, and then co-creating as you're driving forward, that will help align the incentives a lot better to ensure that there is that safety and scientific rigor, from start to end of the process. Because otherwise, we have that enzyme paper, that discovery,
Yes,
things like that will show up. So yeah, that absolutely makes sense. I know one of the things, and this is true for like the protein data bank as well, is that, there's a lot of large data sets now, and they are heavy. They are heavy with so much information, and big models as well that lend this air of authority because, the bigger the better. I think that's something that we have just ingrained into our heads. But when does scale become something that is misleading rather than reassuring to you?
Yes, I think scale can often be misleading
Mm.
because, so one is just when the data is low quality. And I should say, like, machine learning is very good at dealing with data that is has random noise in it. But when you have systematic biases or systematic errors, that's not something that machine learning can necessarily recognize as well. So that's an issue. When particular types of data are missing, also when the underlying paradigm is incorrect of how you're kind of understanding. And so I'll give an example. This is from the early in the COVID pandemic. So there was an app that was used in the UK called Zoe, and it already existed as I think a diet and nutrition tracking app. And so they modified it for people to track COVID cases. And so, in some ways it's impressive. They rolled this out quite quickly, in 2020, early 2020, but they really designed it around tracking a short-term respiratory virus. And so I had questions geared towards that. And so what happened though is that people were developing long COVID, and in many cases were trying to log their symptoms in Zoe, but it wasn't designed for this, and so it didn't list, like the respiratory symptoms. There's like a multiple choice that's easy to check. People that were developing neurological symptoms, fatigue or brain fog, none of that was preset. So they're like hand typing it, and then it wasn't really designed for people experiencing long-term symptoms, so they're having to like retype out their symptoms every day. And so this ended up then being used though in research studies to try to talk about the prevalence of long COVID. And they made these assumptions like, oh, if people stop using our app, it means they've recovered. And I have to credit the researcher Hannah Davis for she's the one who kind of surfaced this case study that's quite disturbing. Whereas it's like, well, this is not what the data was designed for at all. And if you use it for this kind of totally different purpose, where it wasn't easy for people to track symptoms, they got kind of very misleading results because it was the wrong data for the problem they were using. And so that's a concern where collecting more of that, or scaling the way Zoe was deep, and it was very widely used in the UK, but scaling that to even more people wouldn't improve it. It needed a different design and to be collecting different types of data if you wanted to look at this different set of questions with it.
That's a really interesting example because even today, long COVID is something where there are multiple different symptoms coming up depending on which strain you have and whatnot. So I can imagine as they were collecting that information, it becomes very diverse, right? Because each person is
Yeah.
experiencing it in a really different way. And so it's great that you have collection of, information from such a wide group of people and like so much information coming in. But to your point, this is where perhaps a large data set such as this can become a little bit chaotic or noisy when you want to start leveraging, for instance, like a model on top of it, right?
Yeah, and particularly when you lose sight of the fact of kind of no matter what data you're gathering, like there are decisions that go into what's the design of how you gather that data and what you include and what you don't, and what questions you ask or what tests you give people. And those shape what the data set's going to be like. And I think people often are kind of just thinking like, oh, data is objective truth. And it's like, well, it's constructed through this series of decisions that really matter. And another kind of interesting point, or I think important point that case study brings up, is that in that case, there actually were a number of patients who were trying to reach out to the researcher who runs the Zoe app and saying like, hey, this actually isn't meeting my needs at all, or like I've stopped using it for these reasons. And that feedback was not incorporated into the app. And so that's another, to me, really highlights the importance of listening to patients, because they are, have a firsthand perspective of kind of, hey, this is what I'm experiencing, and this is how this tool is not meeting these needs.
I love it. I love the call out for, Yeah, really just meeting the patients where they're at and meeting their needs. And that's so great that they actually receive feedback back from patients who are like, hey, we don't want to use your app, because a lot of times that feedback loop doesn't get generated. And so that's amazing to hear. Also, we know that, as more and more folks start using AI and the modeling for the work that they're doing, if incorrect predictions are coming up and they get published or fed into databases, right? They're not just sitting there, then they become training data for the next model. And I that's one thing that we worry about quite a bit, of the type of error propagation that can seem like a innate structural risk rather than a one-off mistake, because the moment it makes an incorrect prediction and you start feeding it into a database, which then goes into a model, well, then that
Yes.
will continue and that will continue. And at what point can you even find where that mistake was made, in that entire.
Yes, yeah, yeah. And I worry this happens with diseases that are underdiagnosed or where there are diagnostic delays. Of then, often it's seen as like, well, that disease is rare, so it's less likely. But it's, it's also there are diseases like lupus where I think the average time to get a diagnosis is like 6 to 8 years. I mean, it can be quite long, but it's the data that's out there that's getting fed into a lot of these models. Yeah, and as you said, you can get these feedback loops that then can kind of become self-reinforcing.
Absolutely. So I guess, like, in that vein, if we are thinking about teams that genuinely want to reduce the harm that happens to patients, right? Within AI for life sciences, what fundamental practices do you think have to change, even if that means folks need to just move more slowly, which I know is counterintuitive to this whole messaging that we're hearing about AI, which is like, hey, we're going to move a lot faster. But yeah, like what practices need to change fundamentally then?
Yeah, no, I love that you bring up moving slowly because I think it's important. Like all these things of moving slowly and thinking about constraints are really useful. And going slowly to do things well can be quite valuable. Yeah, and so I mean we've touched on several of these, but I think it's really important that we continue investing in research that focuses on underlying causal mechanisms as well, and that as I said, kind of not everything. So our current AI systems, what they're really doing is a kind of fuzzy interpolation between existing data points, and that can be really valuable. And that can also, because it's this fuzzy interpolation, that can also appear novel, but they're not going to give us something truly outside the scope of what the training data was. And so I think that we really do need to still be doing research on areas where maybe we need new paradigms or need to think about different causal mechanisms. Actually, I'll cite here, there's someone who, Arijit Chakravarti, who he's worked for quite a while in pharmaceutical development at all stages. And he has this really interesting idea of the Franken-cell. But he talks about when people pull together pathways from all these different papers, which is something, that we like doing with AI and mathematical modeling. But in some cases, you can end up with these diagrams that would actually never all occur in a single cell. It's like they take this pathway, and he's looking at cancer research of these kind of gene changes that can happen in cancer. And so there are examples where people then publish like, oh, okay, you've got this pathway. And it's like, well, even though each arrow on the pathway is correct, they wouldn't all happen in the same cell. And that's, so he calls that a Franken-cell. And that's, to me, some of the temptation with AI. It's like, oh, it's great. You can throw all these different results together, but really thinking about the underlying mechanism. And in that case, he's pointing out the need to really think of cancer development more as an evolutionary process with randomness, as opposed to this kind of like a circuit diagram. It's going from this arrow to this. So that's one area. I think as I said, kind of continuing to invest in bench science and the development. I know someone who's trying to develop more accurate assays for T cell binding, and that's going to give us different results than just working with the T cell data that we already have, which is also quite biased and only covers a limited segment of the population. But then there are even questions of kind of, well, how accurate is that T cell binding data? So yeah, kind of developing those new assays and new tests. And then a lot of what we've talked about is this kind of this meaningful ongoing collaboration between people with the domain expertise, with the kind of at the different points in the data collection process and the data processing, and the model development and the patients and the people impacted and doctors who might be using it, but really, really making sure that you have that collaboration is crucial too.
Absolutely. So what I'm hearing from you is if we hear of a scientific, pharmaceutical company that's just being run by agents today, then that is probably a big no-no, and we should probably have humans, in the loop who are definitely playing a role in the discovery work that is happening. So yeah, that absolutely makes sense. Rachel, one last question for you before we, hand it over, perhaps a few questions from the audience. What, and perhaps you've touched upon this already, but what has been a really interesting or innovative use case that you've seen of AI within the life sciences world recently? I know there's so much new research being done, so many new use cases popping up. I would love to hear, is there one that's really stood out to you as you've been reading?
I mean, it's something that I find fascinating is this question of T cell binding. And so that's something I've really done a deep dive on. And it's interesting to me in that it's a field where there's still a lot of work that needs to be done. And there even is a competition. It's not quite annual, but they are hosting kind of these regular competitions around, but they're still kind of it's been going on for a few years. I think I would say in the maybe earlier stages of they keep kind of making changes of exactly how they want to frame the competition, which I think is awesome that they're really, trying to thoughtfully think about how do we frame this so that people can really be contributing research and pushing forward at the edge. And so I do like the thoughtful use of competitions and really thinking about how do you structure this correctly and kind of what data do you use to try to spur on innovation, because that is something we've seen. We talked about AlphaFold. Another example is AlexNet arising out of a competition. And so that, and again, that was also, that competition had been running for years. But the way that I think well-structured competitions can push on innovation is something that still excites me. And it's also, I think it often forces people. So my kind of big caveat is, I think AI can be very useful. It's just really important to be quite clear on, okay, like this is the data I'm using in this model. These are the constraints. This is the data that wasn't collected. These are the biases that are going to exist in my data. I still love Timnit Gebru's data sheets for data sets paper about, just really being specific about kind of what data was collected, what are the appropriate uses of it, and then what are uses that would be outside the scope of like, well, actually this data wouldn't be able to tell us about this totally other thing. And I think when you're using machine learning kind of within those clear parameters, it's quite valuable.
Totally agree. Totally agree. I feel like that is a lot of what we repeat over at Can Think Tank. We're like, one, slow down, right? Like, yes, there's a lot that's happening, but we slow down to move fast. Like
Yes. Yeah. No, I love that. Yeah. Yeah.
Yeah, absolutely.
Or go slow to go far is the other saying I've heard. Yeah.
Yes, I love it. I love it. Keeping domain experts in the loop, we need them like from the get-go. That's super important. And then, data is the currency. And so making sure that that data is useful, that you are inputting the right information into the model, but then also training it on those negative use cases as well, right? Like what data isn't here? What isn't available? What mechanisms are we not considering? Having that also listed out really concretely can help.
Yes,
also.
yeah. There's an artist / researcher, Mimi Onuha, who had a really really neat concept and exhibition on missing data sets, where she would like explicitly list, these are the data sets we don't collect and the types of data we don't have. And I think it's really important, to kind of make that explicit and think about what we don't know.
Absolutely. Well, Rachel, this has been so interesting. We have about 9 minutes remaining. And so I think we have time for about 2 questions. So I'll take a look at the chat. I see one from
Great.
Paul. So Paul is asking, what are some of the ways that companies should be adjusting how data is captured and cataloged across assays from different operators to facilitate reliable training of ML agents and models from internal data sets?
That's a good question. I mean, so definitely be recording the metadata and including all that about the about the different operators or different facilities to make sure that that's in there. Because yeah, I know there have been cases of with X-rays or scans where basically they were taken at different locations and that ended up totally biasing the results. And I think part of that is if you don't include that metadata, you're definitely more at risk of that happening. I mean, some of this is going to get into kind of more specific knowledge that I wouldn't have about those particular assays. And so I don't know if there are ways to try to calibrate across the operators that would be helpful of, specifically seeing if you can run the same sample in two locations and comparing those results, but definitely at a minimum to be recording that data and including it in your model.
Yeah, and having that metadata really honed in on and understanding exactly what you're using it for, I think that's going to be a key component as well as you're taking a look at those assays. Great. Any other questions, folks? Go ahead, put it into the chat. While we're waiting for a question, I'll ask you a question that I've been wondering. Rachel, your background is so interesting, and I know you touched upon, studying math and then having this interest and coming into biology. And I guess as, a lot of folks that we work with, they're looking to upskill on new topics. And so, for you, I think it was like going from AI into biology. For a lot of people, it's biology going into AI. Like, what is like a technique or a tactic that you would recommend to upskill in these really hard, topics that we're trying to learn today? Anything that's worked for you?
So I mean, this is hard because I have disclosure. I co-founded Fast AI. Now I work at Answer AI. So, with Fast AI, we ran a series of free courses, which I think still have very valuable content on AI and deep learning. And now with Answer AI, we run paid courses, but I think they're very high quality content that's related to AI. Within the solve, and it's, I should say it's related to a style of problem solving where you use AI but really break things down into small pieces that you can understand, and the importance of really kind of keeping yourself in the loop as a thinker and making sure that you understand what you're doing as you use AI, and that's something I really believe in. Yeah, the biology side, I went through a variety of Coursera and Udacity and HMX and tried kind of all these different platforms. And I did end up going back to school too. And I mean, I found good stuff kind of in lots of places. A lot of it is going to depends on your particular interest and your kind of what you're trying to learn. But there is a lot of great material out there. And it's, yeah, often, and you're finding the balance of, if you try a course and it's just really not the right fit for you. And sometimes that's even just about, where you currently are. And, it's either too easy or too hard. But if you find something that's a reasonable fit, then just kind of sticking with it and going for it. I think there's a lot you can learn.
Amazing. We see a few more questions coming in. So Holly is asking, are there any interesting AI tools that we should know about?
Oh, I mean, this is sorry. My biased answer was I think Solve.it, which I'm working on, is really neat. I'll briefly say it's a Jupyter notebook, basically like a Jupyter notebook, which I think is really appealing and familiar to many people working in data science. That you can then kind of be running AI prompts within the notebook. And it's got some really neat features too, where you can actually edit the output of the AI prompts, which is something that I love because it sometimes when you kind of get into an argument with an AI, then it's got like all this kind of polluted context of things that it got wrong, and this kind of you can just directly edit it, which is neat. But it's kind of pushing you towards trying to keep you in the loop and actively engaged, and not just going off and building huge solutions for you.
Amazing. We'll do one last question just due to time, and that is from Teresa asking, can you elaborate on why you find TCR binding an innovative use case for AI?
Some of this is just my excitement about T cells because I think they're so fundamental to the immune system and just how also realizing the role that the immune system plays in so many different illnesses that I wouldn't necessarily have previously thought of as immune illnesses, but that it's just such a kind of fundamental system in the body. So that is a large part about the value of the application, as well as to me just also that it seems like a problem that we should be able to make big advances on, but that kind of haven't quite got there yet.
I love it. Well, I see a lot more questions in the chat. Don't worry, folks. We see them. We're going to record them, and we will definitely get back to you with the answers to those questions. So keep a lookout for LinkedIn posts, newsletters, articles, whatnot. With that, I just really want to thank Rachel. Thank you so much for taking the time to chat with us. We have really, honestly, I've learned a lot just like through chatting with you just now, and you and I have chatted multiple times already. And every single time I have the privilege of speaking to you, I learn something new. So thank you so much for joining us today. And again, I've posted Rachel's blog onto the chat, so please take a look at it. There are some incredible articles that she's written, and answer.ai, I know that is your new venture, and definitely check that out as well. And with that, folks, we will conclude our session today. Again, if you want to stay up to date on our events, we host an event every single month. We do specific topics for folks who are our members as well, office hours, whatnot. So join our membership if that's something you're interested in, and hopefully we'll see you in March. So thank you everyone for joining. Thank you, Rachel, again.
Oh, thanks so much for hosting. Yeah, this was a lot of fun.
Amazing. Thank you all. Have a great evening.
Thanks. Bye. Bye.